The first thing is the prerequisite for being involved in this foundation is you have to have a daughter named Chloe. Both Kim and I have chosen wonderful names for our daughters. But I did actually want to firstly start by congratulating the organising committee on a really wonderful lineup of speakers today. It's been really informative, and particularly for the mother and infant, a little bit scary to hear about all the things that may happen to your child as they become an adult. And because of that has given me even more drive to get this foundation going. So here's my Chloe, and my journey started about 16 months ago. And like a lot of you in the room who have their own story about the delivery of their child and the diagnosis and all the trauma that goes alongside that, for me coming out of that, I thought, well, how can I put my skills to use? And my job in my career is to solve problems. And normally I'm solving problems for big corporations, but I thought, here's the best problem I could ever put my mind to. Not that I want the problem, but seeing I've got it, where better to put my mind to. So I decided that I had a look around and I tried to find, at first I thought, well, I could just be an extra pair of arms and legs for researchers in Australia and just help them get some grants and that kind of thing, or whatever I could do to help. And eventually I realised that we needed to bring into being an organisation that's really going to focus efforts, raise funds in Australia, focus the efforts of scientists and get the best scientists we have with putting their minds towards the problem of Prader-Willi syndrome and support the ones that are already doing it. But the foundation's not just about one little girl, it's about lots of children, and here's Amelia and Maya and Chloe, and it's also about the ones that are yet to be born, because no family deserves this. Okay, so we see ourselves as one of the pillars that are working for you, working for the families. You've got great work being done by the existing family associations, support associations. You have the Better Living Foundation working on housing, and I can't see James, but he's doing a fantastic job along with the rest of the board there. And then we're trying to be the research arm that supports and delivers outcomes for you. And so with that in mind, we will be talking to you as families, trying to understand what your key priorities are. I mean, obviously to us it looks like hyperphagia and the mental health aspects associated with Prader-Willi syndrome, but we do really want to understand your perspectives on what are the most disabling aspects of this syndrome that we need to make big inroads into quickly. What we want to do is sponsor a very focused program of research. We believe we're going to probably be capital constrained. We're yet to start fundraising, and we have some great people supporting us in that fundraising, great expertise. But given that there's only so much money available, we need to be very focused and have in where we invest our money and make sure there are real outcomes for all of you. We also think it's really important that we mobilise the national Prader-Willi scientific and clinical community, and to that end, we held a conference yesterday in Melbourne, and we brought in some of Australia's best scientists on genetics and on the brain, and we brought in the best Prader-Willi researchers, scientists, clinicians that we have in Australia. And we brought in a couple of people from the US. Fortunately, Dave was already here in Sydney on sabbatical, and he's going to talk to you very soon about some of the very interesting and hopeful work that he's doing in the US. Yesterday was really about working out what our program of research should be, at least putting the first thoughts around that and what our strategy should be in terms of research, but also, as I said, to build this community of scientists. Now, not long ago, this was just a list of names to me. When I say not long ago, I mean October. These people really are the best minds that are working potentially in areas where we can learn something, but not Prader-Willi itself. So Professor John Mattick runs the Garvan Institute. He does a lot of work in RNA, and as Ellie Smith was telling you earlier, RNA is an important part of the genetic puzzle around Prader-Willi. Tony Hannon, who is at the Florey Institute for Neuroscience and Mental Health. Martin Pera, who is Mr. Stem Cells Australia. Rob Nichols, who was here earlier but no longer, as Ellie mentioned, he's working on a pig model. So there are problems with the mouse models and in getting them to reproduce the full set of Prader-Willi syndrome. I won't go through everyone here, except to say we do have Dave here. Ellie was there yesterday, and she's been here today. Stuart Einfeld provided some great contributions yesterday. And of course, Joe came along as well. So we've had, and we did have one other person I should mention, Dan Grant, who is from Pfizer, and he can help us understand how to quickly get any potential compounds into market. So at that stage, I'm going to hand over to Kim, who's going to tell you about some of the key messages that came out of yesterday's workshop.
Thanks, Kath. And I think it was a real privilege yesterday to be in a room with so much brain power and to look at all the expertise and the skills that they were bringing to put their mind to thinking about how we might come up with breakthrough treatments for Prader-Willi. And as Kath said, most of them had no real understanding about Prader-Willi syndrome, but that's actually often good because you can bring in and have new insights into how we might move the field forward. And I came away with several key messages that I thought were just important to briefly share. And I think one of the first key messages came from John Maddock in his opening speech. And what he said was that clinicians give the care, which is very important, but researchers give the hope. And for me as a researcher, I don't need to be convinced about the importance of research. And so, but I think that really emphasises why we need to do research so that we can have better treatments in the future than we currently have. The second key message that I came about was if we believe that research is really important, which I think we all hopefully all do, how are we going to excite the researchers in Australia to do Prader-Willi research? There's only 1,000 or so people with Prader-Willi in Australia. It's not a major public health problem. There's not many researchers working on it. So how are we going to excite researchers to bring their skills to address this problem? And researchers, they are driven by, and they need to get high impact publications, they need to get money to fund the research, and that all goes on their track record. So how can we excite them? And I think again that came from something that John Maddock said when he was describing the genetics of Prader-Willi syndrome, and we've heard quite a lot about that today, and that region on chromosome 15 where the problem is, that gives us Prader-Willi syndrome, is actually the window to the brain, because that region controls so many systems throughout the body. And so what I saw was that many of the researchers that were in that room were excited about that possibility, because what he said was if they can understand what's going on at that region of the chromosome, then they will understand a lot of all of how the brain works, which will then have implications for a whole range of other treatments, and that excites researchers over and above the Prader-Willi. The other aspect that I came away with again is that if we want the researchers then to do the research, we need to get funding. And we know that funding is very difficult to get. How are we going to convince the NHMRC, the government funding agency, they want to fund major public health problems? How are we going to get them and philanthropists and so forth to fund research into Prader-Willi syndrome? Unless we have a very wealthy person, a family who have a person with Prader-Willi syndrome, such as Michael J. Fox, you can see what happens when you have someone who is really wealthy or high profile, you can get a lot of funding happening. How do we do that without having someone in that space? So to get the funding, what are we going to do? And again, I think what I took away from it is that we can use Prader-Willi syndrome as a model to solve the answers to some of the society's major public health problems. So what the story was, again in the room, was that using Prader-Willi as a model will really help us to understand the genetic mechanisms of obesity and the major public health problems such as the psychoses and anxiety and so forth. And so again, that should be our push when we go to seek funding. Yes, it's important about Prader-Willi, but it's over and above that. What we want to be pushing is that use Prader-Willi as a vehicle to understand and to provide insights into these other public health problems. And then we're much more likely to get funding from industry, from drug companies and so forth to support our efforts. The other key message that, again, was really reinforced by a lot of the speakers was the critical role that families and people with Prader-Willi syndrome play in driving the fields forward. And that's not just in Prader-Willi, but in any other condition, that it's the families and the people with the condition that are really the pivotal people that allow it to happen. And there was many examples given of a family member, a mother, a father, who had taken on the call, had no experience in science, and had come up with the answer. I mean, I think we've all seen this, Lorenzo's oil, all of those examples where it took someone with drive to do that. And we will talk a little bit just at the end about how we can all be mobilised to help that. And just before Dave talks, and then I'll just stand up at the end again, but just to acknowledge Kath for the enormous effort that she has put in, her own resources, her own funds, her own time, giving up her consulting work, paid work, to set this up. When she first spoke to me about it, I said, yes, I'd be happy to help. I didn't realise that she would be able to bring together, and as that list of people, I don't know if you know, but that is the who's who in Australian research, and for her to be able to bring those people together to give up a day of their time is, I think, an extraordinary effort, and I just want to thank you for all you did there. So now we're just going to hear from Dave about his work that he's been doing that really has a lot of implications for Prader-Willi.
All right, thank you very much. Yeah, I'd also like to thank Kathleen Jones for allowing me to come and talk to you today about the work that I'm doing. And I think what I want to talk to you about today is just to give you an idea of some potential avenues of kind of therapeutic development targeted for Prader-Willi research. So when I think about therapy, I could kind of think about two ways of going about it. One is to try to start with maybe what you call the top-down, which is you see symptoms, you see hyperphagia, you see anxiety, you see psychosis, you see hypertonia. These are all things that you could see, and then you say, well, how do we make a therapeutic that could treat those things? But as we've heard from the previous speaker, sometimes that's not so easy. These are complex phenomena, and sometimes we could come up with good drugs to do something about that, like human growth hormone, sometimes not. Another way of looking at it is what you might call from the bottom up, which is, what are the things that we do know on a molecular level? And can we try to address those kinds of things, and maybe that will have some clinical benefit. So one of the things that we know about the disease is something about the genetics. And I think you've all heard about genetics today, and certainly Professor Smith has talked about genetics a lot just recently. So I think that everyone in this audience is familiar with this kind of gene diagram, and the fact that in Prader-Willi syndrome, there's usually a large loss of the genes on this paternal region of this paternal chromosome. And then all of these genes in red here are off, and this gene is still on. But something about these genes being off is kind of fundamentally what initially causes this disease. And so from a very simplistic point of view, which is where I come at this, the question would be, can we somehow turn these things back on? And would that have a benefit? Now, if this kind of deletion occurs on the other chromosome, that leads to Angelman syndrome. And in the case of Angelman syndrome, which is the area that I've been working in for longer, we have the other situation where these green genes are now on, but this gene is off. And this gene seems to be off because of this long non-coding RNA that I'm sure you're all familiar with. So it's making all the things that it needs to make over here, so that this person does not have Prader-Willi syndrome, and these genes are also on, but this one is off. And so for Angelman, the therapeutic goal was to see if we could somehow reactivate this gene, which is now off, in the brain, and see if we could turn that back on. Now, the reason that it is off in the brain is because of this long RNA, and in some way, this RNA going in this direction prevents this gene from going on in the other direction. No, let's not do that. So So what we'd like to do is, in the case of Angelman, is to come up with some way of turning this gene on either directly or by stopping this RNA that is somehow inhibiting this gene from being expressed. So that's kind of the simplistic goals that we start with. You know, how do we get this gene back on? Now, one of the ways that we can think about doing that is say, how does this work in bodies in general? So there's a lot of things that go into gene regulation. There's a bunch of different kinds of RNA, there's DNA modifications, and there's also proteins that play a role in this. And some of these proteins come along and they contact the DNA right near the gene, and they tell the gene to be on or tell it to be off. We call them transcription factors. And so the kind of approach that my lab has taken, and certainly this is not the only approach by any means, but the kind of approach that my lab has taken is to say, well, can we learn how these natural transcription factors turn genes on and turn genes off, and can we then design something that can turn on just this gene? And how could we even think about doing that? So I'm just going to take a little aside and just tell you about a recent development that allows us to do that. And that is this area of genome engineering, which largely relies on these targetable nucleases. And so I think one of the greatest revolutions, at least in genetics in our time, has been the development of these targetable technologies. And without going into a lot of detail about what these are, let's just say that these are proteins that we can essentially reprogram to go to specific genes and do something. The things that I'm showing you here are set up to be nucleases. So they go by names like zinc finger nucleases, tau nucleases, and CRISPR. I think Dr. Smith mentioned CRISPRs very briefly. And these are proteins that can be targeted to have an action at a specific gene, a gene that we can decide which gene that's going to be. Now, for the nuclease, and this is just to give you some examples of where this technology is heading. For the nucleases, there's a couple of things that this allows us to do. Directing a nuclease to a particular spot on the DNA can allow us to make mutations there in a targeted way, or it can allow us to correct mutations that are already there. So just to make some concrete examples of that, there's a protein on the surface of cells in the blood that is required for the HIV virus to bind to those cells and infect. And if you could somehow mutate that protein on the surface of those cells, the HIV virus would essentially just bounce off the surface. And so some scientists have developed this as an approach to HIV therapy, where they target the gene that makes this particular protein that HIV needs to bind to, and they used one of these targetable nucleases to target mutations to that gene so that now it does not make a good gene product. And essentially they can make cells that the HIV virus cannot infect. And so the way they do this therapy is they take the cells out of the patient, they modify them using this targetable nuclease, and now they're making HIV-resistant cells, and then they introduce them back into the patient. And hopefully those cells will go on and regenerate an immune system. And just last year, they released the results of a clinical trial. So this is a targetable nuclease that's in clinical trial. And they were able to show that this is actually something that reduces the viral load in people with HIV in their clinical trials. So that's kind of, an example of trying to create genetic mutations. And, another example of trying to correct genetic mutations, and just one example of many examples I could give. We've heard that, there are some people that have mutations in their MAGEL2 gene that seem to have Prader-Willi-like phenotype. And so in principle, one could go in with one of these targetable nucleases and set it up in such a way that you could correct that mutation back to wild type, what we call wild type or normal gene, and then they would have that function back. Now, that sounds like a great capability. If we could develop that as a therapeutic, that would be wonderful, and there's certainly many, many people that are trying to push the science in this direction, in addition to me. So we're going to see this coming, but we have to also think about the many challenges that there are in this kind of thing. So I described to you how they did this HIV therapy, and they had one major advantage that we don't have in this case, which is they could take the cells out of the body, put them in an environment where it's easy to manipulate them, and then put them back in the body. In our case, we're maybe looking at a gene that's in the brain, and we just can't take the cells out of the brain and change them and put them back. So we have to bring our thing to the brain, and that's not an easy thing to do. But that was the challenge that our lab faced when we tried to come up with an approach to turn that gene on in Angelman syndrome. So we're not working with nucleases, we're working with a different version of these proteins. These are just going to be proteins that turn genes on or turn genes off. And in the story that I'm going to talk about briefly, we designed a protein that could turn a gene off, and it's going to try to turn off this transcript, this long black antisense transcript that is repressing this UBE3A gene. And what we hope to see here is if we put this protein into a cell or into the neurons in the brains of a mouse, that it will turn down this black antisense transcript and turn on this UBE3A gene. So I'm going to summarize about 5 years of research in our lab in one slide. People in my lab would not like that, but let's just say we designed these proteins and we designed them in a way that we had hoped that we could inject them, purified protein, into mice, that they would go and cross the blood-brain barrier and get into the cells in the brain. And so this is, I'm just showing you some, this is my only data slide. I'm just showing you some data here where these are slices through mice that we treated. And the blue is just a stain that shows you kind of where the cells are. And then there's a red dye anytime the protein is present. So you could see that in these two mice that were injected either with this therapeutic protein that we call S1 or this other one, you can see a kind of red glow there, hopefully. Whereas in this non-treated wild-type mouse, there's not really a red glow more than background. And in this mouse that's a model for Angelman syndrome, meaning a mouse where this the maternal gene is not expressed, we also don't see that glow. Now the question is not whether the protein gets into the brain. The question is, does the protein get into the brain and turn that gene back on, that UBE3A gene? And so in this one, this is the wild-type mouse that expresses UBE3A normally. This is the Angelman syndrome mouse down here that doesn't express UBE3A because it has a mutation. And here, when we put on our therapeutic protein, you could see that now the mouse looks more like a wild-type mouse and less like this Angelman mouse because it's expressing UBE3A in its brain. So again, in summary, what we've done here is we've created this protein to target this region of this gene. We can turn down this transcript, and that turns the UBE3A back on. And we think we can do this in the brains of living mice. So that's what our data is showing us. Now for Prader-Willi syndrome, again, we have the kind of opposite situation. We don't want to turn this thing on, or we don't want to turn this thing off, but we want to turn it on. So if we could take the same factor, this was the kind of simplistic idea we had again, if we could take this same factor and now instead of making it a factor that tells the gene to turn off, what if we make it a factor that tells the gene to turn on? Maybe we could turn on this antisense transcript here. It might not affect the things out here, but at least all the things that are in here should go back to being green. That was our simple hypothesis. And so this is a project that's funded through Foundation for Prader-Willi Research in the US. And I'd like to tell you the punchline of this story, but this is experiments that are in progress now, so I can only tell you to stay tuned. But I would just, again, I think the purpose of me telling you this story here today is just to give you an idea of different types of research, different types of genetic tools that are becoming available, and work that's going on that eventually we're going to start to see where this line of research is heading, and hope to see if we can go from cells to mice to test animals, and then hopefully see these kinds of therapies enter clinical trials. I don't have any more time to tell you about any of this work today, but we did publish a paper recently where we talked about some of the challenges of this work, in terms of specificity, toxicity, and delivery of these kinds of molecular therapies in the brain. So I would encourage anyone interested to take a look at that. And then I would just close by thanking the people in my lab and the funding organizations that supported this work, and thank you.